Bellows is one of an ongoing series of in-depth reports on research progress in the areas of the natural sciences, agriculture, and the environment, produced for the College of Natural and Agricultural Sciences at the University of California, Riverside. This is the Riverside Report.
There are a few more chairs if people want to get going or to have a seat. We are going to start. I'm Steve Engel. I'm the Dean of the College of Natural and Agricultural Sciences here at UCR. It's my honor to welcome you to this grand opening celebration of our new insect.
On May 4th, 2001, faculty, administrators, and interested community members gathered at the University of California, Riverside to dedicate a new building, the Insectary and Quarantine facility. Aside from the speeches, there were insect displays and photo ops and tours of the new facility for members of the press. At a time of rapid growth in Riverside, this particular building has attracted more than the usual level of interest. UCR's entomology department is indeed world-renowned. Established in 1910, the department has retained and enhanced its reputation over the decades, in great part because of the use of biological control rather than chemical pesticides to eradicate insect pests and diseases. Today, more than ever before, exotic and dangerous insects threaten agriculture in California, as these ravaged vineyards attest. Mark Hoddle from UC Riverside is working to eradicate this threat using the classic techniques of biological control developed at Riverside.
When the sharpshooter showed up in California approximately 10 to 12 years ago, it was found in the southern part of the state, and it has slowly moved from this initial area of discovery, probably on nursery plants which have been moved from Southern California into the more northern parts of the state. Now, in some areas where the glassy-winged sharpshooter has established, it's been associated with the decline of grape-growing industries in those areas. And this is primarily because of its ability to spread this bacterium which causes Pierce's disease. This bacterium lives in the xylem of the grapes, which are the water-conducting tubes inside the grape plant, and as the bacteria multiplies, it essentially clogs the plumbing of the plant, and water can't move through the plant anymore, and scorch-like symptoms appear, and essentially the plant dies of dehydration. So some of the major control strategies for the glassy-winged sharpshooter initially focused on chemical control. That is the use of pesticides to try and suppress sharpshooter numbers to levels which wouldn't cause damage to grapes or other commodities. Some big problems have surfaced with the excessive use of these chemicals. One is groundwater contamination from nursery areas as the pesticides run off has become a major concern with the Department of Pesticide Regulations. We're also concerned about the sharpshooter's ability to develop resistance to these insecticides. That is, it will become tolerant to these poisons and they'll no longer be effective. To mitigate these potential problems, we were asked at the university by the California Department of Food and Agriculture to initiate a biological control program. So when we initiate a biological control program, that is when we go back to the home area of the target pest, we prospect for these natural enemies. When we find the natural enemies which attack the pest, we bring them back to an insectary and quarantine facility. So essentially the quarantine facility is a safe harborage for these new exotic organisms which we are bringing into California. Once we have the natural enemies in quarantine, we run safety tests to make sure that they attack only the target organism, and we also screen them for their own suites of natural enemies. The new insectary facility which we have at Riverside is a state-of-the-art facility. It's the best facility available, probably the west of the Rocky Mountains, probably as far as a public institution goes, maybe in the entire United States. The facility has improved our ability to do biological control work because it provides more space for us to do our experiments, more rearing space for the mass rearing of these natural enemies before we release them, and the facility is more secure than the old facility that we currently have at UC Riverside. Biological control is the deliberate importation of exotic organisms by scientists to control an exotic pest. So Essentially, we are going back to the home range of the pest. We look for what we term host-specific natural enemies. We don't want to be releasing into the environment an organism which could become a problem itself.
Peter Atkinson, another faculty member at UC Riverside, believes that he too is practicing biological control, but in a way that is both new and exciting and somewhat controversial.
My training was a geneticist, and I've always been interested in genetics, and ever since I was in high school, and insects have always provided an ideal way to study genes, and entomology on top of that provides a way to not only learn about how these genes work, but to actually use that knowledge to basically help with real-world problems. In this case, agricultural problems, and in the case of mosquitoes, serious medical problems that are some of the most serious problems in the developing world. What I work on here at Riverside is basically developing gene transfer technologies for insects of medical and agricultural importance, and what we're doing at the moment is focusing on ways to introduce genes into mosquitoes, specifically those mosquitoes that are disease vectors for diseases both in the US and overseas. The definition of biological control is a very interesting one. Biological control can be a very broad category, and under that category can come the term genetic control. Some people will like to differentiate between the two. Some people will lump them together. In my opinion, I think in a sense we're splitting hairs. What we're seeking to do is to introduce new genes into species either through genetic engineering or through classical breeding, and then further down the track, once those particular organisms have been tested in the lab to make sure that they're safe, releasing them in the field in order to reach a solution about some problem out there. And that, in a sense, is not a lot different than classical biological control, where you've introduced not a new genetic strain in our case, but a new species altogether into the field in order to really achieve the same outcome. The way that we make insects transgenic is the same way that scientists have been making Drosophila transgenic for many years, since 1982 in fact. What we simply do is that in the lab we use standard recombinant techniques to make these small DNA molecules called plasmids, into which we can clone many different types of genes. And we inject these plasmids into the specific parts of an insect embryo. Insect embryology is very different from mammalian embryology, and it enables us to target those particular nuclei of the developing embryo that actually give rise to the next generation of, in our case, mosquitoes. So all we simply do is take a very fine needle. The bore of the needle is less than a millionth of a millimeter in diameter, and we insert that very gently into the posterior pole of the embryo, and we can tell, depending on the species, which is the posterior pole. These embryos tend to look like drawn out footballs. And we use air pressure to actually insert the solution containing the plasmid DNA into the posterior pole of the embryo. If we've been clever and careful enough with these injections, most of these embryos will survive. They'll become larvae, they'll pupate, they'll become adults. And what we do then is simply mate these adults with our standard laboratory strain of mosquito. One peculiar aspect of this work, and one crucial aspect, is the type of actual genetic vector that we're using to introduce the gene into, in this case, the mosquito, or it can be a fruit fly, it can be a moth. We use transposable elements, which are small pieces of DNA which are ubiquitous amongst all organisms in which they've been looked for. And these transposable elements are called jumping genes, and they occur naturally, and they have the ability to move from one particular piece of DNA into another piece of DNA. And all we're really doing in the lab is augmenting this natural process by using these transposable elements to insert our genes of interest into the insect, in our specific case, the mosquito. One of the most exciting things about the progress in the last few years is that we've really only had the ability to introduce genes into mosquitoes since about 1998. And in that short period of time between then and now, a number of labs around the country and around the world have been able to put in a number of genes which on paper should produce a mosquito that has reduced ability to transmit disease. But it's worth remembering that these particular beneficial genes that researchers are putting into these mosquitoes are ideas that have been circulated for 5-10 years now, and in a sense they've been sitting in the laboratory refrigerators or freezers as ideas waiting for this dam to be burst, this dam being the ability to put genes into mosquitoes and other economically and medically important insects. And the progress that's been made in this short period of time has been heartening and stunning at the same time. The real excitement is that this is just the tip of the iceberg. We know so little about how pathogens, whether they're viruses, whether they're parasites, protozoans, or worms, get transmitted through mosquitoes. For this to occur, the pathogen must first be ingested by the mosquito, and it has to then pass through a number of barriers in the mosquito, through the gut, through the hemocoel. It has to evade the mosquito's immune system, and then has to get into the salivary glands, and then be injected into uninfected individuals in order for the parasite or the pathogen to complete its life cycle. It's also worth remembering that many of these diseases that mosquitoes transmit are what we call resurgent diseases. Up until the 1970s, they were basically under some level of control around the world, but since then, with the decrease in the use of insecticides, many of these mosquito diseases are now coming back and threaten the borders of the United States. And we desperately need new solutions in order to control these resurgent diseases. And the ability to really go in and understand how these pathogen genomes, these mosquito genomes interact with each other really presents us with a unique opportunity, which this new facility will enable us to capitalize on, because when we develop new hypotheses to test, we can actually make these insects here. We can test them here with this new facility.
Tom Bellows from UC Riverside made quite a name for himself in the 80s by applying biological control methods to the invasion of Southern California by the notorious ash whitefly. He headed a committee of faculty and staff in the specification of all the components of the new facility that make it safe and effective for advancing research into exotic and transgenic insects.
We're standing today outside the new 2001 Insectary and Quarantine facility at the University of California in Riverside. This facility is a new world-class facility to allow us to continue our research on natural enemies of insect pests in California and other parts of the United States. Let's go inside today and take a look at some of the facilities and all that they can do. Now, the primary purpose of this building is to safely and securely bring in natural enemies into California, where we can test them before we release them into the environment to ensure they're going to be safe and not harm anything. Now, that happens when natural enemies are shipped in boxes like this. They're brought to a highly secure room through airlocks like this. Now, the airlocks are black because insects fly towards light, so insects will never fly towards an airlock. Now, once we get into this room, this is the most secure room in the facility. It's called a primary quarantine facility. And inside these shipped boxes, we will find containers containing natural enemies. Now, natural enemies are typically shipped in very small containers. Here I have about 24 natural enemies, and they're very small. They're hiding inside these vials. And natural enemy work in quarantine is typically done in teams. Now, Andrew's going to take care of running every one of those vials through that microscope and record what she finds, while I would sit down and then fill out the paperwork for where they came from, what country they're from, what host plant they're from, everything the collector or the overseas collaborator sends us in terms of information. One example of how this works is a few years ago, we went to Mexico and brought back two natural enemies, Incarcia and Idioporus, that attack a whitefly that's killing hibiscus plants and other subtropical plants here in California. Now, here in this room, what we do is once we get them out of these little vials, we give them their favorite food. We give them the pest insect to eat. Now, here you can see, and we keep these in cages, of course. Here you can see is a citrus plant, and the undersides of the leaves are just covered with these little insects that are just devastating. In this case, this is a devastating insect pest for citrus, and we're working on a natural enemy for it. And when the natural enemies come in, they stay in this room for a generation or two. They have to be tested for diseases. They have to be sure that they're not going to harm any other natural enemies that we have around. We try to be sure that they attack this pest and attack it very effectively. Once we've proven that, even though they're kind of confined in small spaces here, we can then move them to rooms where we can rear them into larger numbers. Those are called quarantine rearing rooms, and we're going to take a look at one of those right now. Now, as we exit the primary quarantine receiving room, that's where we first open and work with the bugs. We then work to rear more of them, but we're still inside the quarantine boundary, which is this wall. And outside each is a suite of quarantine rearing rooms that has negative air pressure to it. By the way, that's so that if whenever this door is open, bugs go in, nothing can get out. And these suites of rooms then give us an opportunity to rear larger quantities of bugs, to do the experiments in the field, to try and get California the best benefit it can. These rooms are essentially just cubicles. They have temperature control, humidity control. They will be filled with racks that will have cages of plants on them, secured to the wall for earthquake safety. There are rooms like this in which the lights can be turned on and off all by themselves. Clear at the other end of the hallway, we find rooms that have also daylight. Now, some insects are very fussy about things, and they actually won't mate unless they see daylight. So about 1 in 5 rooms in the facility has natural light as an option. And again, this room has the same features, the temperature and humidity, as all the rearing rooms do. Inside the quarantine barrier, you may be wondering why all the walls are painted black. Well, under normal operating conditions, the lights are off except these ultraviolet lights. Now, these ultraviolet lights attract insects that might have by chance gotten out here, and they get stuck into sticky material along the top and bottom of this light trap. And that material is cleaned and investigated every day by the quarantine officer. It's just one more security measure. Negative air pressure, dark hallways, a light to attract insects, so that nothing can get out of here. Here we are in between two of the working floors of the building. This is called an interstitial floor. It houses all the ducting, the electrical, the communications, all the laboratory gases and vacuums and pressures. It's where all the controls are for separate temperatures for every room in the entire building, and between each of the 3 working floors is one of these interstitial spaces. The material and operations is so complex, we had to provide separate space for all the workings so that the research can get done on the floors themselves. Now, in the quarantine floors, of course, this is especially important because all the maintenance can take place without the maintenance people ever having to interrupt or breach a quarantine protocol. On every floor, we have an environmental chamber room, similar to those rearing rooms before, but they've got much more sophisticated controls. In these rooms, we can change the temperature every minute, every hour. They also can get much colder than our other rearing rooms. So if we have to bring something from the southern hemisphere in the winter, we can simulate that winter here and bring them through to our spring quite rapidly so that they can match our climate. Nothing is permitted out of the quarantine boundary except living, healthy insects that we know will benefit California. Everything else, everything that comes from overseas and everything we work with, gets sterilized, either through an autoclave or through a dry heat hot box like this one that I'm going to walk through and come out the other side. Voila, things that come out this side are completely sterile and harmless, and they get thrown away.
This building, the Insect and Quarantine facility, is particularly significant to us. Not only will it support our teaching and research programs in very important ways, as you're going to hear shortly from the chair of the entomology department, it's also important to every Californian.
What has been done to bring us to this stage is prelude to that which will occur here, and that's infinitely more important. The promise that this facility, and more importantly, the people who will work in this facility have for the future of our university and our state is mind-boggling, and I think well worth celebrating. It's 14,000 square feet. I won't tell you how much it costs per square foot.
It includes 2 receiving rooms, 6 research laboratories, 12 greenhouses, 64 rearing rooms at biosafety levels 1, 2, and 3. Each room has its own climate control, temperature, humidity, and lighting that can mimic climatic conditions hospitable to insects brought here from all over the world. Between levels of containment, as you will see, is a series of airlocks and decontamination rooms. You will have the opportunity to see for yourself the intricacies and complexities of this remarkable structure, truly state-of-the-art. And as you saw in your insert for your invitation today, once it's open, it's closed. We will have licensed individuals only who will have the ability to gain access to the facility. Currently, a new pest not known before in our state enters every 60 days. And this is liable to accelerate with increased travel and trade. Every year, these exotic pests cost California agriculture over $3 billion in lost production. The Insectary and Quarantine facility will serve the $26 billion state agricultural industry by developing biological control and other alternative pest management strategies to deal with these invasive pests and diseases. But the work conducted in the facility is not limited to agriculture. The facility also protects California's urban and natural environments by developing techniques to manage pests that cause untold damage to our structures, landscapes, and forests. It protects the public health by facilitating studies on such threats as mosquitoes and Africanized bees. Research conducted here results in economically and environmentally sound alternatives to pesticides, thus reducing the chemical load in our land, air, and water. In a sense, this facility touches every aspect of our lives: our home, our health, our environment, the food we eat, and if you remember the pesky ash whitefly, the very quality of our lives.
Century. The question has been frequently posed to us: what can you do in the new facility, and why is it important? The first answer is more of the same, and that's not a trivial comment. Biological control has been one of the key strengths of the Riverside campus, even before it was a campus. Scientists associated with our department have traveled around the world and have returned with predators and parasites to control pests that have been introduced into the state. In recent years, we've had more pests and more potential projects than we've had space in our current facility to accommodate. With the addition of the new facility, we've eliminated a major bottleneck to acting swiftly to solve problems. The building also provides our scientists with the capability of conducting research that was not possible in the past. As you'll hear on your tours, we can conduct research on the interactions of insects and genetically modified plants, and allow those plants now to go to flowering and to the fruiting stages. The level 3 quarantine spaces have air filtering systems that trap pollen and bacteria and not allow them to escape into the environment. As the Chancellor mentioned, we can conduct research also on genetically modified insects in the facility. Finally, we now have the capability, the security, and the capacity to conduct research on insects not found in California and find solutions for potential problems before they enter the state. For example, we have some of the best chemical ecologists in the country in our department, and they now have a facility to conduct research that can provide better tools for detection and trapping of potential pests. These tools can be deployed to prevent pests from becoming established, which is clearly the most efficient strategy for avoiding pest problems. The faculty in our department have trained the outstanding students who have taken leadership positions both nationally and internationally. This new facility will allow us to continue to attract and train the best. As you can tell, I'm very proud of my colleagues and our exceptional staff who have given so much of themselves to make this day a reality. Please join us for a tour of the building. I'm sure that you'll see why my pride is justified.
The canister which I'm going to be opening contains 2,000 ladybird beetles, commonly known to people like me as ladybugs. Most of you also know it's an effective biological control agent, a generalist that we welcome in our home flower beds and vegetable gardens. but it's much more than that. It's a close relative of the Vidalia beetle, which was the very first success story in biological control. In the late 1880s, the cottony cushion scale threatened to wipe out citrus production in California. The USDA scientists traveled to Australia and brought back the Vidalia beetle, which he had observed feeding on the scale. The beetle was reared and released through Southern California. The experiment was a great success, resulting in such a dramatic reduction of scale that the citrus industry became a strong proponent in biological control. To this day, for well over 100 years, the Vidalia beetle has kept the cottony cushion scale under control in California citrus. To commemorate that first success and to symbolize the future successes which we hope will result from our new Insectary and Quarantine facility, I'm going to release a relative, one that we're familiar with, 2,000 ladybird beetles.
As this symbolic release of one of Riverside's earliest successes, the common ladybug, attests, the new Insectary and Quarantine facility represents just another step by generations of scientists at UC Riverside to develop, perfect, and apply the methods of biological control of dangerous insects and diseases. But it also represents the future. Some of the claims we heard at the dedication might have seemed hyperbole or the natural exaggerations of an institution's pride, but in this case, they weren't. This facility does have the potential to protect vital industries like agriculture and even save lives, perhaps millions of them.